So a HOX gene is only one homobox gene, and it only occurs in animals, and it allows for the position of the body parts of the animal, and it only occurs in clusters, whereas the homobox gene is similar genes, direct for the location of the parts of the body, and once again, so basically every HOX gene is a homobox gene, but not every homobox gene is a HOX gene. So this can be seen as in examples of the fact that animals are commonly segmented. The backbone of vertebrae, or rings in a worm, have specialised over time for functions. So, as you can see here in the insect, that it's segmented, but so is a worm. A worm is segmented, but for their function. So, we've got 3 different types of symmetry here that you can see in animals that can express and prove this HOX gene formation. So we have radial symmetry. Radial symmetry, you can find the colour, is seen in dioplastic animals, so such as jellyfish, little jellyfish legs here. They're called dioplastic, and that means they've got 2 primary tissue layers, dioplastic, or plastic, whatever. So that's radial symmetry. Asymmetry, which you can see in red here, asymmetry is in a sponge, so you have no lines of symmetry. And we have bilateral symmetry. Which is in like majority of animals, which has the left and right sides of the head and the tail. So the HOX genes can prove, so the bilateral is basically every animal and is the core example to prove the HOX gene formation of the segmented animals, so the head and the tail and the left and the right, so left and right sides of the head and the tail. So the HOX genes with mitosis and apoptosis. So if you imagine some clay work, someone who is making clay, to produce a final shape, the clay maker as such takes the clay away and adds more during the process. So there's taking the clay away and adding some more clay during the process of making the actual product. So this is basically what happens in the production of making somebody. So the mitosis is obviously the making more cells to leading to growth and the masses of the enlargement of the animals. So the mitosis is extremely important, but also apoptosis is really important in making the human. So is the termination or cell death. So cells undergoing apoptosis often release a chemical signal that stimulates the mitosis and the cell proliferation. So an example of the development of a morphogenic apoptosis is a hand. So the mitosis is the fingers on the hand, but the apoptosis is the skin between your fingers that are taken away when you're an embryo. So I just have here the run through, get this over, of the stages of apoptosis. So you start with a pre-apoptotic cell. The pre-apoptotic cell is here. So it starts with the nucleus condensing. After the nucleus condenses, the cell shrinks, as you can see here. The cell shrinks and the nucleus actually fragments into smaller parts, as called nuclear fragments. And then you get apoptotic bodies engulfed by a phagocyte, which is, you get here apoptotic bodies which have nuclear fragments in them, and they are engulfed by a phagocyte. The cells undergoing the apoptosis often release a chemical signal stimulating the actual mitosis, which is the making more of the cell, and cell proliferation. So it's not just cell death, it is actually encouraging more cells to be produced in mitosis. So we get the cell nucleus condensing. So firstly, nucleus, my little green nucleus. The nucleus condenses. the nucleus condenses inwards. So this is the actual nucleus. The nucleus condenses, the cell in itself shrinks as the second step. So the whole cell just shrinks, goes smaller. The cell shrinks, the nucleus fragments, so you get nuclear fragments in a late apoptotic cell. You get an apoptotic body is engulfed by a phagocyte as the last, so they're not just floating around the cytoplasm. You do also get the membrane blebbing, it's called membrane blebs. So that's when the membrane, when it is the cell is shrinking, during the cell shrinkage. The factors affecting the expression of regulatory genes, so we're going back to expression of the genes in the operon, etc., etc. These factors expressing the regulatory genes includes the change in temperature, so the change in temperature, the light intensity, internal factors, especially for multicellular organisms such as hormone release and phagocytosis, psychological stress. So stress and release of the hormones actually change the expression of the regulatory genes because of the hormones. And these factors have a greater impact during the growth and development of an organism, so teenagers and embryos, because obviously you're still maturing and your body is completely changing. These factors have a greater impact during growth, but drugs can significantly affect the regulatory genes. An example of this is formaldehyde, which was to prevent pregnant sickness, prevented normal expression of a HOX gene, so the babies had shortened limbs. It can now be used actually in cancers to prevent the networks of capillaries for tumours developing, so it kills off the tumours. But that is an example of how drugs can affect the regulatory turning on and off type of genes.